Hello, everyone. Thank you for staying with us through this virtual event. My name is Luciana Lim. I'm the Regional Marketing Manager at Thermo Fisher Scientific, taking care of the Southeast Asia and Taiwan region. We have a very special panel prepared to tackle topics ranging from ever-evolving landscape of cell and gene therapy development to partnerships within the ecosystem and how informed decisions around manufacturing models can help developers achieve their clinical and commercial milestones. I am joined by our three panelists, Dr. Sylvie Shen, Mr. Li Shing-Mou, and Ms. Chen Bingrong, who has shared much about their work earlier on. Hi, Sylvie, Shing-Mou, and Bingrong. Good afternoon to you all.
Good afternoon.
Hi, good afternoon to you all.
To everyone.
We know that in Asia, at least 20 CGT products have been approved by local authorities for domestic use in the region. We are also witnessing a growing pipeline of novel therapies, as well as a surge of innovative solutions, technologies, and players coming onto the scene. Even so, ongoing challenges like cost, scalability, regulations, and lack of standardization in the field continue to be a potential roadblocks in achieving commercial viability. Let me start the ball rolling by posing this question to Sylvie and Shing-Mou. From your experience working and collaborating with therapy developers, what are some of the common challenges that you have seen them struggle with, and what kind of support were you able to provide to alleviate these problems? Maybe could I have Sylvie first to answer it?
Thank you, Lucy. We've seen progress of CGT manufacture often hampered by multiple factors. That includes the limited selection of raw materials, the high cost of production, such as viral vector production in the case of viral-based CAR T-cell manufacturing, and also the lack of fit-for-purpose instruments. All these all contribute to the time, resources, and ultimately the costs that are required for such development. At Thermo Fisher, we have developed cell therapy system or CTS branded product to help address some of these challenges. Our CTS branded product are GMP manufactured, specifically designed to meet the needs of clinical and commercial manufacture with appropriate regulatory documentations. And CTS branded raw materials such as media and reagents, they comply with the latest guidelines for ancillary materials, and the product undergoes vigorous safety testing that includes mycoplasma, endotoxin, and sterility, and comes with supporting traceability documentations. And CTS branded viral production system, such as LV-Max and AAV-Max, they offer scalable and cost-effective solutions for viral productions. And also our CTS cell therapy instruments, including DynaSelect, Rotea, and the Xenom electroporator, and their associated single-use consumables. Like the CTS media and the reagents, all CTS products undergoes extensive safety and validation testing. It complies with regulatory guidelines and includes regulatory documentation packages that helps for customer filing. And we have dedicated regulatory and quality team to provide product support for CTS branded products. And such products have been used in over 200 clinical trials globally. Thank you.
Thank you, Sylvie. It's quite reassuring to hear that Thermo Fisher actually provides a whole range of complete workflow for cell and gene therapy with the CTS branded solutions. What about Shing-Mou? As a researcher in CGT field, what would be the common challenges that you face, and how did you face these challenges?
Regarding the living drug, because CGT product is a living drug. Living drug means metabolic active and with capacity to reproduce in vitro. And the most important thing is the after injection administered into human body, the product still remain its function. So both the biological and the chemical physical testing method used for characterize the living drug. It is the very complex because biological is the biological characteristic is the central of the characterization. It's based on the assumed MOA, and due to the study materials and the complex mechanism of action, it's to develop a reliable and quantitative bioassay is the most challenge for CGT researcher. Now, for example, as I mentioned in my talk, the inhibition of T cell proliferation is the most popular bioassay used for measuring the immunosuppressive capacity of MSC. It's a very important assay, can directly measure the effects of the MSC on the T cell proliferation. However, the assay system remain include two living cells. The first one is the effector MSC and the responder T cells. So the interaction of the two cells is a bi-directional. It's a very complex mechanism, and so it is very hard to handle to control control and validate the assay. So that's the reason why mobile developed a quantitative ideal assay to replace the inhibition of T cell assay for measuring the immunomodulation capacity of the MSC cells.
Thank you, Shing-Mou. Yeah, thanks for sharing your perspective on the drug potency analysis portion and give the audience some examples when it comes to the testing. I would like to follow up with another question for yourself. Among the different bioanalytical tools to support the preclinical and clinical studies of cell and gene therapies, I'm curious as to how well digital PCR has been adopted in the CGT applications. Would you like to share more on this?
For me, yeah, I think recently the development of the CGT products is expanding rapidly in Taiwan. So the authority, Taiwan FDA, just released guidance for the strategy for development of CAR-T products in this year, February. So it recommends the developer of the CAR-T product and need to develop the analytical method to characterize the CAR-T product, and in the early stage, and must use all kind of the analytical method to characterize the CAR-T products. So the digital PCR can measure absolute quantity. So it can be used for safety and the identity and the potency analysis. I believe the precision medicine and the precise analysis is the trend, and digital PCR is one of the important tools for GDT development. That's my comments.
I actually completely agree with you that precision is actually of utmost importance in supporting the development and safety application of cell and gene therapy, especially with the increasing adoption and shift towards targeted therapy. Now, I actually wanted to hear from Sylvie regarding the adoption of Thermo Fisher's QuantStudio Absolute Q digital PCR and CGT application, considering that it has been in the market for a few years now. Sylvie, would you like to share?
Sure. Yeah, we have seen a strong 12% compound annual growth rate in the digital PCR market, and Asia Pacific has been the key growth region at 15%. And in fact, the QuantStudio Absolute Q digital PCR system by Thermo Fisher has seen over 600 installations globally, with over 30% of them being adopted in the biopharma sector, despite being newly launched in the last 2 years. We are just in our third year now. One of the key benefits for CGT workflow when comparing digital PCR versus conventional qPCR is the elimination of the standard curve in the test. Digital PCR is able to perform absolute quantification directly. The standard curve is not required, and taking out the standard curve significantly reduces data variation and increases the accuracy, precision, and reproducibility, which ultimately makes the whole workflow more cost-effective. In addition, digital PCR also gives you that extra boost on sensitivity when you are looking at low copy numbers. And last but not least, your workflow as in the steps in performing digital PCR reaction is not going to be any different than qPCR. So the transition is very easy to implement as well.
Thank you, Sylvie, for sharing that transitioning to digital PCR is not difficult at all. This information, I'm sure, will benefit our audience who are currently using qPCR, as it puts them in a better position to consider switching to digital PCR to achieve the higher sensitivity, which is a requirement for cell and gene therapy applications. I would like to now shift our focus and hear from a regulatory perspective, right? Earlier on, it was mentioned that selecting quality raw material is critical, especially when developers want to move quickly through the phases of drug development. I would like to include perspective from Bingrong here. What are the key factors that organizations should consider when selecting critical raw materials to cater to both early and late phase requirements? Bingrong?
Thank you. So starting with the GMP raw materials in early phase will actually help to reduce the comparability studies during the late phase development. So some companies may actually use the non-GMP raw materials in the early phase because they are cheaper. But it's actually crucial to actually work with the suppliers early to fully understand if these raw materials will still be able to comply with the late phase regulatory requirements, or there's actually a need to switch to a comparable raw material. It's also necessary to actually understand the full set of documents that is available to support the customer's following needs. Our recommendation is that suppliers should be able to assist companies to select the most suitable raw materials in order to fulfill their needs. So some of the raw materials can be actually customized. So it's necessary to approach the suppliers at early stage to discuss on the options that's available to meet your customers' needs. And authorities also tend to focus on safety aspect of the raw materials, which is actually as early as a phase 1 R&D stage. So for critical raw materials that contains animal or human-derived components, viral activation, clearance, as well as validation studies would be required. So all in all, for critical raw materials with proprietary formulation concerns, it's also necessary to consider discussing with the suppliers at early stage on how the suppliers will be able to assist in obtaining such information. So for countries that accept the raw material master file, as earlier has mentioned in the presentation, this can be an effective way to present to the evaluators. And for regions that do not have this master file system, also consider discussing with with the suppliers as well as sometimes with the evaluators on how we can better support in this form for clinical trial needs. Yeah, thank you.
Right. Thanks, Bingrong, for sharing that. And you brought up a good reminder to work with suppliers of raw materials to ensure that they actually know how to support you, work together to provide the necessary regulatory support files. I actually have a follow-up question for you. Specific to the Southeast Asia Taiwan region, can you also briefly comment how developers can reduce the risk in their CGT development in developing countries with less defined guidance?
Okay, so risk in CGT development actually lies in the raw material selection and qualification, the CMC data, and analytical characterization. The team actually has earlier mentioned the potency as well as comparability. So collaborating with the regulators at early stage is also encouraged because it helps to ensure regulatory clearance for such human studies, especially when limited animal models and preclinical data are available. The regulators' experience can also provide valuable insights into establishing safety considerations and influencing the developers' approach in their study. So in addition, it's also necessary to speak to suppliers to select the well-characterized raw materials without the animal origin components, and also to ensure that there will be adequate technical documents that will be available to support their filing needs. This is actually some of the ways to help to reduce risk when it comes to CGT applications. The use of the well-characterized GMP raw materials will also help to reduce the unnecessary changes and comparability studies. Bioforum white papers also offer some guidance on leveraging these platforms and process characterization data for CGT validation, as well as guidance into raw material risk classifications as well as assessment. So these are actually some of the documents that customers can actually gain into access to find out more information regarding the best practices around CMC as well as process establishment, which can help to accelerate the CGT development. Strategic planning for robust data to meet the country's most stringent regulatory requirements should be started as early as possible, taking into factors on the countries that the companies actually want to venture into and launch their products in the latest phase of commercialization.
So based on your earlier insights, you mentioned that the use of well-characterized GMP raw materials can avoid unnecessary changes in comparability studies. We know that Thermo Fisher's Ginkgo CTS portfolio of cell and gene therapy products are GMP manufactured, safety tested, and backed by regulatory documentations. Can you elaborate on the regulatory documents that are available and how users can actually request for them?
Yeah. So documents available for the CTS products will include the user guide, the certificate of origin, which is also known as a COO, certificate of analysis, the COA, the regulatory support file, which is commonly we call it as the RSF, and the drug master file. So for equipment system, we actually have the same amount of document support with additional validation guides as well as the certificate of conformity. The RSF and the DMF are actually requested via a web link, and the RSF can be provided to customer under a CTA that's been signed with Thermo Fisher. We have recently also submitted the DMF for CTS Frontier System to US FDA, which is in compliance with ISO 23565. So to better support customers, we have also released a CTS Equipment System Validation Guide that provides customers with an overview of the type of validations that's performed on our equipment system. So this will actually help the customer to do their self risk assessment and also to demonstrate to the authority the level of conformance that we have on bioprocessing systems. which is in compliance with the international standards and the regulatory requirements. Yeah, thank you.
Right. Thanks, Bingrong, for sharing such a comprehensive list of available documents that supports our users. I believe those are readily available on the web, or everyone can reach out to us to get more information about the mentioned documents. Right. So besides regulatory compliance, actually raw material considerations, as mentioned earlier on, deciding a commercially viable manufacturing model is actually very critical to success. Developers often grapple with questions around whether to perform activities in-house or to outsource, centralized or decentralized manufacturing, and whether to invest capital in a certain process platform in the market. The implications of these decisions can actually be very long-lasting. So, Shing-Mou, what advice would you have for therapy developers as they consider their manufacturing model?
Yes, to introduce a new manufacturing model is always difficult for for a lab decision maker. As as a lab head, I not only the cost and the flexibility and the scalability and the regulation compliant. I also consider the long term after service and the providers long term after service and the capacity capability of the maintenance and the troubleshooting for the manufacturing models and from the service provider standpoint, I think the outsourcing is a good alternative for a developer in the at the early stage because you don't need invest huge resources on facility and the instrument. Only focus on developing your core tech. And Mobile has experience to provide the regulation compliant manufacturing and testing service to assist CGT developer to conduct their clinical trial in Taiwan and in overseas. Thank you.
Thank you, Shing-Mou. There's really a lot to consider for a therapy developer. Sylvie, what about you? What advice would you have for therapy developers as they consider their manufacturing model?
Sure. Yeah. When choosing a manufacturing model, there are several factors to take into consideration. Your core competencies and the cost implication that may help with the decision for your in-house versus outsourcing manufacturing, and also consideration for scale and flexibility. Determine if your manufacturing model can accommodate future growth and increase demand, and also consider the flexibility required to adapt to evolving regulatory guidelines, and also the technological advancement and the market dynamics. And centralized manufacturing may offer economies of scale, enabling large production volume and potentially lower costs per unit. And decentralized manufacturing, on the other hand, can provide more flexibility to adapt to regional or local market. Likewise, in an all-in-one manufacturing system, integrates all process steps in one single equipment setup that can offer more streamlined manufacturing operations. However, it often has limited scalability and lacks the flexibility to accommodate changing parameters in the process. Modular system, on the other hand, consists of separate units or modules that perform specific function of the manufacturing process. This approach offers flexibility to scale production by adding or removing modules based on the demand, allows for customization and adaptability, as different modules can be configured to meet specific process requirements. And our CGT system is such a modular system that offers flexibility and scalability. And our CTS Cellmation software provides a flexible system to managing and integrating these multiple instruments to streamline our cell therapy workflow. And lastly, collaborations with experts. Engage with industry experts, consultants, and experienced manufacturing partners to gain insights into best practice or lessons learned, to leverage their knowledge and experience to make informed decision about your manufacturing model, and to help maximize the chances of success for your cell and gene therapy development. Thank you.
Thank you. Thank you, Sylvie. I believe I should have time for the last question. So finally, to actually help overcome challenges associated with manufacturing and scaling, Thermo Fisher has also established a partnership program to assist customers on their path to clinical and commercial success. So Sylvie, may I invite you to share more about the Collaboration Center program that is available in Asia Pacific as well? What are the benefits and supports that collaborator of Thermo Fisher can be expected to gain from there?
Yeah, sure. And we believe collaboration and partnership is instrumental to the advancement in cell and gene therapy field. And our Cell and Gene Therapy Collaboration Center offers Cell Therapy Initiative or CTI program that aims to accelerate the development and commercialization of novel therapies by providing access to Thermo Fisher's experts and technologies and resources. And our partner can expect to gain several benefits and support, such as access to the state-of-art technologies and infrastructures for research and manufacturing, expertise and guidance from our team of scientists and specialists, assistance in progress development, optimization and scale-up of manufacturing, as well as regulatory support to ensure compliance. And ultimately, this will fast-track your journey to commercialization. Just as in the example, in addition to the collaborative projects I mentioned in our talk earlier, we also ran a successful collaborative project with Prescient Therapeutics. Prescient is a clinical stage oncology company that focuses on the development of a universal CAR-T platform. And through the CTI collaboration, they've been able to adopt our CGT workflow and leverage supports from our expert scientists, and that enables Prescient to close their process and fast-track their process development for scaling up to a clinical relevant scale. Thank you.
Thank you. Thank you all for such an informative discussion. There are some great key takeaways that I would summarize. So the Thermo Fisher Skipco CTS portfolio is comprised of a fit-for-purpose products across media, reagents, and cell therapy instrumentation, as well as viral vector systems. And these products are GMP manufactured, safety tested, and backed by regulatory documentation to support customers along their path to commercialization. Several of the CTS products are already in use in clinical trials and commercialized therapies. Second, digital PCR offers unparalleled sensitivity and accuracy, enabling precise quantification of rare genetic targets crucial for cell and gene therapy applications. Its ability to detect low abundance target with high precision empowers users to make informed decisions, ultimately advancing the development and optimization of cell and gene therapy treatments. Finally, we encourage you to have conversations early with your supplier to make sure that they have documentations to support your regulatory filings and the capability to scale up manufacturing to meet late phase and commercial demands. Perform a raw material risk assessments and prioritize aspects that are critical to your business. I would like to thank our panelists, Dr. Sylvie Shen, Mr. Li Shing-Mou, and Ms. Chen Bingrong for their time and valuable insights. Last but not least, I would like to take this chance to do a little advertisement here. In the upcoming Singapore Cell and Gene Therapy Pan-Asia Summit 2024 that will be held in Singapore from 18th July to 20th July, Thermo Fisher Scientific is proud to be the diamond sponsor of the event. The theme for this year's event is Enabling Cell and Gene Therapies in Asia. So this conference brings together the CGT leaders in Asia Pacific like yourself to share groundbreaking R&D, practical insights into how CGT can enhance preventive health and patient care. Most importantly, fostering strategic partnership. If you're keen to join us at the event, feel free to scan the QR code here on the slide and register. I would like to now officially close the panel discussion and thank everyone for listening in and submitting your questions. If there are any questions we could not address on today's call, we will review it and try and follow up with you as appropriate. Thank you, everyone. Now I would like to hand it back to Israel.
Well, thank you again to everybody who stayed with us through all of our wonderful webinars today. As a reminder, these talks will be available for on-demand viewing shortly. So I've been Israel Gonzalez, and I'd like to take a moment to thank Labroots and Thermo Fisher Scientific for making this event possible. Although we've concluded our presentations for today, this event is far from over. There is a survey found in most of the rooms and also the top navigation bar, so please take a moment to fill that out, as we'd love to hear your feedback. We encourage you to continue to explore the virtual environment, chat with your peers, check out the many resources that are available in both the lobby and the networking lounge, and also visit the virtual lab. Until next time, everybody, goodbye.
Goodbye.
Thank you.